guys, welcome to Becoming Dr. Kaluba. It's your girl Faith, and I'm back with another video. So today I am giving a research seminar at Andrews University. Am I nervous? Yes. Yes, I am, but I'm keeping a prayer in my heart. I am so excited for this very full circle moment. I attended Andrews University for my undergrad, and so just being able to come back 2 years after graduating to talk about my research and to speak to other students who are doing the biology program and the biochem program, different array of programs that they're studying. It's amazing. It's very humbling. It's exciting. It's just, I'm really appreciative of the opportunity. So I thought to also have you guys join me as I give my research seminar talk. So yeah, see you soon. Let's get it.
Good morning, everyone. Welcome to our October seminar here in biology. It's a pleasure and a joy to bring back to our campus one of our alumni. Faith comes to us from Grand Rapids, where she currently works as a cancer biologist studying metabolism and those kinds of things at the Van Andel Institute. So she's here to tell kind of her story, not just the science, but kind of what her journey's been like here at Andrews and beyond. So Faith, welcome, and thank you for coming.
Thank you so much. Good morning, everybody. Thank you so much, Dr. Zdor, for the introduction and the invite, for allowing me to come speak and share my research with you. Thank you to Dr. Lyons as well. As mentioned, my name is Faith Kaluba. I am an assistant research technician in the Leon Lab at the Van Andel Institute. And today I will be sharing with you my research on beta-hydroxybutyrate metabolism in cancer cells. One of the core findings of my research is that beta-hydroxybutyrate is preferentially used for fatty acid synthesis in cancer cells. Now, as many of you probably know, cancer is a disease characterized by abnormal cells that divide uncontrollably and destroy body tissue. It is also the leading cause of death globally, according to the World Health Organization, who conducted a study in 2020 looking at new cancer cases, where they found almost 2,500,000 new cases of cancer across a vast array of the different types of cancer. So this has created urgency for many scientific researchers to enhance our understanding of cancer and improve treatment outcomes. One of the outcomes of the numerous studies investigating cancer and its mechanisms is the identification of over 6 hallmarks or features of cancer, one of them being metabolic reprogramming. Now, metabolic reprogramming defines cancer cells' ability to adjust their nutrient usage to promote their proliferation, survival, migration, and differentiation. It is also driven by cell intrinsic factors like lineage. Just one second, let me get my pointer on. Like lineage, genetic mutations, and signaling that shape the metabolic network. Now, recent findings suggest that extrinsic factors such as the tumor microenvironment also play a role in influencing a tumor's metabolic network. Our lab specifically is interested in defining how the tumor microenvironment regulates the metabolic network through metabolite availability under physiological conditions. Our knowledge of this would potentially offer insights for diet-induced therapies targeting cancer metabolism. Now, our hypothesis is that changes to the diet that then contribute to changes in the plasma nutrient levels will in turn influence nutrient levels in the tumor microenvironment, thus altering cancer metabolism and tumor growth. The intention, again, is to provide a basis for potential synergies between dietary factors and new or existing therapies targeting tumor progression. Interestingly, there's already a wealth of literature available on different diets touted as cancer cures that is readily available on platforms like Amazon or your local Barnes and Noble bookstore. However, many of these proposed remedies lack substantial scientific research supporting their claims. Intriguingly, a common trend observed among these diet suggestions is nutrient restriction in some capacity. Whether it's the methionine restriction diet or the cysteine restriction diet or others, all propose that limiting one or more nutrients hinders cancer growth. And we have already established that depletion of certain nutrients in the tumor microenvironment can influence cell dependency on other nutrients. In recent years, there has been increasing research focused on two specific dietary approaches that reduce glucose availability to the cells. These are the calorie restriction diet and the ketogenic diet. Now, both diets are low glycemic or low sugar diets, which are popularly thought to prevent tumor growth. In the ketogenic mouse diet, for example, there is 90.3% fat, 8.7% protein, and 1% carbohydrates. Whereas the calorie restriction diet, mouse diet, consists of 40% restriction of carbohydrates, 28.4% restriction on fat, and 31% protein, with respect to the control diet, which is made up of over 60% carbohydrates, 18.8% protein, and 17.2% fat. Now, both diets trigger the same bodily response that is observed during fasting. So as your blood levels fall due to low glucose, the pancreas elevates secretion of the hormone glucagon. The secretion of glucagon results in the activation of lipases and the reduction of insulin secretion. Now, these lipases release fatty acids in the form of triglycerides from fat cells, and these triglycerides are then converted into ketone bodies like beta-hydroxybutyrate and acetoacetate in the liver. These ketone bodies are then transported through the blood to surrounding cells where they can serve as alternative fuels for your cell metabolism. Now, while both glycemic diets result in ketone body synthesis, their differing nutrient compositions raise questions about potential variations in their effects on cells, particularly cancer cells. This was the case in an in vivo study on the impact of these two diets on tumor growth. So in this study, what they did was inject a pancreatic cancer cell line in mice subcutaneously. So a sub-Q injection is pretty much an injection just below the skin rather. And then they let the tumors grow up until they became palpable. So just up until they sort of look like a boil or big enough to measure. And then once the tumors became palpable, they switched the tumors on put the tumors on calorie restriction diet or a ketogenic diet, as well as some put on the control, which served as a control. In this study, it was observed that the calorie restriction diet, but not the ketogenic diet, significantly impaired growth, which was measured through tumor volume over a period of 20 to 25 days. So if you look at the graph here, you notice that the tumors on calorie restriction diet were significantly lower than those on a controlled diet. But when you look at the tumor growth of the mice put on a ketogenic diet, you see that there's basically no significant difference between those on the control and the ketogenic diet. So letting us know that there's something different. There's something about the calorie restriction diet that allows for tumors to grow slowly. But when you compare the blood glucose levels, you notice that both diets had the same effect. They both dropped the blood glucose levels in the mice. So this suggested to us that the observed outcome on the left was not necessarily due to the low glucose levels, but probably something else. In the same study, they looked at both the plasma and tumor interstitial fluid levels. I'll call that TIFF. Going forward from the mice on both calorie restriction and the ketogenic diet with respect to the control as shown on the y-axis, where the more negative the y-axis, the lower the levels of the particular metabolite on the x-axis. And the more positive the y-axis, the higher the levels of that particular metabolite. And what they found was that that beta-hydroxybutyrate levels were high in both the plasma and the TIF, which got us wondering what purpose beta-hydroxybutyrate serves and whether it is metabolized by tumors to support their growth and progression. So far, we know that beta-hydroxybutyrate is a ketone body synthesized from triglycerides in the liver. We also know from the mouse study that it is elevated in circulation under both calorie restriction and the ketogenic diet. I should also mention, for those who may have not taken chemistry yet, beta-hydroxybutyrate is a 4-carbon ketone body. So a ketone body is basically an acid that has a carbonyl group attached to 2 R groups. So in this case, your R group is another carbon as well as a hydroxyl group. But what we don't know is whether beta-hydroxybutyrate can be metabolized by cancer cells. We also don't know if all cancer cells are capable of metabolizing beta-hydroxybutyrate. We're also wondering what exactly is beta-hydroxybutyrate used for by these cells, and if our understanding of its use could inform our current insights into the role of diet in cancer progression and treatment. So to address our questions, we began by looking at the beta-hydroxybutyrate metabolism pathway as observed in normal cells. So when circulating beta-hydroxybutyrate enters the cell, it can be transported into the mitochondria, where it is converted to acetoacetate by BDH1. This acetoacetate can then be converted to acetoacetyl-CoA by OXCT1. And this is then converted to acetyl-CoA by a mitochondrial thiolase. Now, you may know already that acetyl-CoA can react with oxaloacetate to form citrate, which then either feeds into the citric acid cycle or exit the mitochondria into the cytosol, where it can be converted back to acetyl-CoA. Acetyl-CoA in the cytosol can then be used to synthesize palmitate following its conversion to malonyl-CoA. So to answer our questions on whether beta-hydroxybutyrate can be metabolized by cancer cells, and if all cancer cells have the machinery necessary to metabolize beta-hydroxybutyrate, we first conducted a Western blot to confirm the expression of BDH1 and OXCT1 in a panel of 6 cancer cell lines. So the AL1376s are a murine pancreatic cancer cell line, murine being mouse. The B16s are a murine melanoma or skin cancer cell line. The Miapacas are a human pancreatic cancer cell line. The A549s are a lung cancer cell line. The HeLAs, cervical cancer, and the PANC1s are a human pancreatic cancer cell line. So yeah, A549s all the way through PANC1s are human, same as Miapaca, and AL1376 and B16 mouse cancer cell lines. We discovered that of our panel of 6 cell lines, only 3 expressed both BDH1 and OXCT1. These are the AL1376s, the B16s, and Miapacas. Now, I should mention, for those who may not be familiar with a Western blot, we also ran or probed for vinculin, because vinculin serves as like a housekeeping protein. So just to control for the amount of protein that you load, to confirm that it's pretty even across the board. So we knew from this point that not all cancer cell lines have the machinery to metabolize beta-hydroxybutyrate, just based on the pathway. Then we proceeded to ask the question, for those that do have the machinery, how does beta-hydroxybutyrate contribute to their growth? Now, based on the composition of the calorie restriction diet and the ketogenic diet and how they lower glucose levels, we thought maybe beta-hydroxybutyrate supports growth of cancer cell lines in low glucose conditions. And so we replicated this by growing all 3 cell lines in both high glucose, that's at 10 millimolar glucose, and 0.1 millimolar glucose. Now, I must mention, these are in vitro or cell culture conditions. Your body will never drop to 0.1 millimolar glucose because homeostasis. But to sort of try to model that in the in vitro setting, as if you notice in the Miyapaka cell line, we had to drop our concentration of glucose to 0.05 because the cells are still able to grow at 0.1 millimolar glucose. So we really wanted to make sure we establish a low glucose condition in vitro. So when we grew our cells under these conditions and monitored their growth as shown by the doublings per day. So the doublings per day are literally what they sound like, how much the cells double each day or how much they proliferate. What we observed is that at 5 millimolar beta hydroxybutyrate, the cells pretty much did not grow as well as they did in the high glucose condition. And going forward, I will refer to their growth in beta hydroxybutyrate conditions as rescue, to mean to restore their growth to what is observed in the positive control, or to observing an effect that's even greater than what we see in our positive control, our positive conditions. So having that observation led us to believe that beta hydroxybutyrate maybe doesn't rescue the cells by supplementing glucose or by supplementing the citric acid cycle per se. As you may already know, glucose is used for the citric acid cycle, to synthesize citric acid cycle metabolites, as well as for ATP synthesis downstream. But what we also know is that glucose can also be used for palmitate synthesis. And so then we decided to test whether it's possible that beta hydroxybutyrate rescues growth of cancer cells in the lipidated conditions, or when lipids are absent, because like glucose, beta hydroxybutyrate could also contribute to palmitate synthesis because acetyl-CoA can be synthesized from beta hydroxybutyrate. So when we did this in all three cell lines, our B16s, AL1376, and Miyapakas, we observed a different result. When beta hydroxybutyrate was added to our minus lipid conditions, or our lipid-starved conditions, we saw that it rescued the growth of cancer cells significantly across all three cell lines. So it got us thinking that maybe beta hydroxybutyrate rescue occurs through its contribution to lipid synthesis. To further confirm this rescue and it being due to contribution to palmitate synthesis, we conducted a proliferation assay experiment as well, where fatty acid synthase, the enzyme that converts malonyl-CoA to palmitate, was inhibited to test whether indeed the observation we saw in minus lipids was accurate. When we did this in B16s, ALs, and the Miyapakas, we noticed that addition of beta hydroxybutyrate to our minus lipid conditions did not rescue the growth of these cell lines, as we had observed previously in the absence of a FASN inhibitor. This confirmed to us that the contribution that we noticed, or the use of beta hydroxybutyrate to support cancer cell growth, was indeed through palmitate synthesis or lipid synthesis. To further confirm our observation, or to use an alternative method to do so, we decided to incorporate the use of GCMS. So as you will probably learn, GCMS contains two components, gas chromatography and mass spectrometry. Now, gas chromatography is the first component. It separates the different components of the mixture. Mass spectrometry then further separates the components based on their mass to charge ratio. So the results obtained from this process include your gas chromatogram, which we use for qualitative and quantitative analysis, and the mass spectra, which we use to identify both known and unknown analytes. So once your sample is placed on the autosampler, it is then injected into the column where the molecules are separated based on polarity and volatility. When the molecules separate, they are detected at 0.7, one by one, and the gas chromatogram is developed. They then enter the mass spectrometer, where they're bombarded with electrons to ionize the molecules and then separated in a mass analyzer, which contains two electrical fields. Now, these electrical fields allow the ions to oscillate based on their mass to charge ratio, and they are detected and displayed in a mass spectrum. Now, the combination of this data allows us to analyze and interpret the label distribution into various metabolites, including citrate, acetyl-CoA, and palmitate. So looking back at the pathway by which beta-hydroxybutyrate contributes to palmitate synthesis, when fully labeled beta-hydroxybutyrate, 13C beta-hydroxybutyrate, is added to the cell, the label diffuses across its downstream metabolites. So notice that beta-hydroxybutyrate does not contribute to oxaloacetate. So citrate is not fully labeled, which then impacts the cytosolic acetyl-CoA label and finally the palmitate label. Now, based on this pathway, starting from citrate to acetyl-CoA to palmitate, if, say, for example, you have 75% of your total citrate pool from the cell labeled, your expectation is that molecules or products that are downstream of that citrate will only be at most 75% labeled. They will be less than or equal to 75% labeled. The same follows for palmitate. Palmitate will only be as labeled as acetyl-CoA because it is downstream of acetyl-CoA. So just bear that in mind for now. But before I proceed into more mass spec data, I want to just talk about labeling and how best to interpret the data. So it follows that contribution of fully labeled acetyl-CoA to palmitate occurs in an even-numbered mass isotopomer distribution. This is because the synthesis of palmitate occurs by addition of 2 carbons from malonyl-CoA. Malonyl-CoA is synthesized from acetyl-CoA to make a 16-carbon compound, which we know as palmitate. Now, what this implies is that labeled palmitate exists in M+2 to M+16 forms, where the number refers to the carbons in palmitate that are labeled. So this is represented in a binomial distribution because, again, the amount of label in palmitate depends on the probability of labeled acetyl-CoA being incorporated into it. What this means is that if we have a scenario where 25% of the total pool of acetyl-CoA in the cell is labeled, then there is a 25% chance that for every molecule of palmitate generated, 4 out of 16 of those carbons will be labeled. As a result, the binomial distribution of palmitate label shifts to the left, with our M+4 having the highest amount of label. The same follows if, say, 50% of your acetyl-CoA pool is labeled, then there is a 50% probability that half of your palmitate carbons will be labeled. As a result, your result will be, or rather your label distribution will be even in the sense that, or central, because you have, again, a 50% chance of label. So it will be more centered around your M+8. The same follows, again, if you have 75% label of acetyl-CoA, you have a 75% probability that 75% of your palmitate will be labeled. So again, your highest peak or your highest isotopomer of palmitate will fall at M+12. And so your graph will be more shifted to the right. So please pay attention to, I ask that you pay attention to the shifts in the graphs for the coming data. I should also add that the shifts in the graph, whether left or right, are indications of how much label or how much label is getting into palmitate. So if you have your graph being more left-leaning, it tells you as well that there is less label getting into palmitate. If you have a graph that is more right-leaning, it tells you that you have more label being incorporated into your palmitate. So we first started by looking at beta-hydroxybutyrate labeling into AL1376s, specifically focusing on our palmitate label, our citrate label, and our acetyl-CoA label. So based on this graph, you notice that beta-hydroxybutyrate fully labels palmitate in our AL1376s. When we looked at our citrate versus acetyl-CoA, so bear in mind, citrate is a 6-carbon compound. So you will have M+1 through M+6 isotopomers or forms of citrate. acetyl-CoA being a 2-carbon compound, you will have N+1 through N+2 labeled forms of acetyl-CoA. Now, when we took a look at our citrate and acetyl-CoA data, our expectation initially was that, again, based off of the pathway, acetyl-CoA being downstream of citrate, we would have less label into acetyl-CoA than citrate. But when we looked at our AL1376 data, we noticed the opposite, where there was more acetyl-CoA label than citrate, which doesn't make sense. You can expect to have more label into a downstream product than an upstream product, unless there is another means by which the label is getting into the downstream product. So bear that in mind for now. So based on that result, we then started to think about how this could be possible. We then went back to our glucose data and thought, okay, let's try to label glucose and see if we get a similar result with what we see when we label beta-hydroxybutyrate. And so we labeled our glucose and traced it into palmitate, and we saw the same result in A. So this is glucose in the absence of beta-hydroxybutyrate, unlabeled beta-hydroxybutyrate. And we saw a similar result where the label gets all the way into N+16 palmitate. But when we compared our citrate and acetyl-CoA data, we got what was expected initially before we conducted our beta-hydroxybutyrate tracing experiment, where the acetyl-CoA label was lower than what we see in citrate. We then proceeded to test what would happen if we traced glucose in the presence of unlabeled beta-hydroxybutyrate. So the assumption is that for any differences in the label, or any reduction in the label, we know that that unlabeled citrate or acetyl-CoA or palmitate was coming from our unlabeled beta-hydroxybutyrate and not our labeled glucose. And when we tested this, we noticed that the shift in our palmitate label had moved to the left, which tells us that there's less amount of label from glucose getting into palmitate. And it also additionally tells us that there's that the cells prefer to use carbons from beta-hydroxybutyrate for palmitate, even in the presence of a substrate like glucose. The same follows for our citrate versus acetyl-CoA data, where even though the phenotype observed in the absence of beta-hydroxybutyrate was maintained, where acetyl-CoA label was lower than citrate, the amount of contribution to both citrate and acetyl-CoA dropped when beta-hydroxybutyrate was added to media with labeled glucose. So this told us again that somehow, even with glucose label present, abundant glucose label present, the cells still somehow prefer to use beta-hydroxybutyrate as opposed to glucose. So thinking again about the difference that we observed initially in our parental AL1376 line, where we had more acetyl-CoA label than citrate label, we then went back to our pathway. We started thinking about where the additional label from acetyl-CoA was coming from. This would mean that at some point during this pathway, one of these metabolites is getting out of the mitochondria and being metabolized there and converted to acetyl-CoA. But we weren't really sure at what point in the pathway this was happening. So we incorporated CRISPR knockout gene editing. So if you don't already know about gene editing, you will come to learn about it. So if you don't understand it right off the bat, there is hope. You will learn about it soon. But I'll just roughly explain how exactly we go about CRISPR editing. So CRISPR is a tool that we use for a variety of purposes. You can inactivate a gene with it, you can knock down a gene, but in this case, we decided to knock out the gene. So what exactly happens is you sort of come up with a vector complex, including your Cas9, and you generate a single guide RNA, which serves as a target for your desired gene sequence on your DNA. In this case, in a knockout, our aim was to delete the particular genes that we wanted to look at, our BDH1 as well as our OXCT1, based on the pathway for beta-hydroxybutyrate metabolism. And so we did that utilizing this vector system And then we transfected our vector in our cancer cells. The vector also contains an antibiotic resistant gene. So in this case, I believe we used puromycin or geneticin. So you want to have an antibiotic resistant gene such that when you do your selection, the selection process is important for you to verify which cancer cells actually took up the plasmid and were successfully knocked out. So you verify using your selection, you add your antibiotic, the cells that die let you know which ones didn't take up, and the ones that do survive let you know that your plasmid was successfully taken up. You grow your cells, let them be confluent, and then you use flow cytometry to sort of develop single clones or single colonies of your cells, which you then expand, expanding meaning growing your cells in different size plates. So the more cells you have, you transfer to bigger size plates. And then to confirm successful knockout, we used Western blotting again, where we probed for BDH1 and OXCT1. So again, going back to the pathway, we decided with first generating knockouts for BDH1 to test whether beta hydroxybutyrate, for starters, was just left in the cytoplasm or could go back into the cytoplasm and be converted there. So we confirmed knockout. The SGBDH1 and SGBDH1 number 1 and number 2 are your knockouts, and your SGNTC is your non-targeting control. So when you're doing CRISPR, you also want to generate a control. Your control is what is somewhat likened or more close to your parental cell line. It is the cell line that goes through the entire process of CRISPR and we assume is unaffected by the knockout process. So you have your non-targeting control where we confirmed BDH1 was still present, and we confirmed successful knockout shown by the absence of a band in our BDH1 blot. We then repeated the same experiments where we grew our cells in minus lipids and added beta hydroxybutyrate. And in the non-targeting control, as expected based on the parental result, beta hydroxybutyrate still rescued the growth of the non-targeting control. But when we grew our knockout cells, both 1 and 2, in minus lipids media containing beta hydroxybutyrate did not rescue their growth. The same happened when we trace labeled beta hydroxybutyrate into both palmitate and citrate and acetyl-CoA. We noticed that in our knockout cells, label did not get into our citrate and our acetyl-CoA in our knockout. Yeah, I just said that. In our knockout, no label got in. But when we looked at our palmitate distribution, we noticed that in the knockout in both plus and minus lipid conditions, the distribution had shifted to the left. That signifies that beta hydroxybutyrate was not contributing to palmitate synthesis when we knock out BDH1. So that told us that that additional acetyl-CoA label that we saw initially that was higher than the citrate label was not as a result of beta hydroxybutyrate somehow going out of the mitochondria and into the cytosol and being synthesized there. If anything, BDH1 was required for beta hydroxybutyrate to contribute to fatty acid synthesis. We then knocked out OXCT1 and repeated the same experiments. We discovered that beta hydroxybutyrate rescues OXCT1 knockout in minus lipid conditions in both of our knockouts, opposite from what we saw in the BDH1 knockout. We also tested the same by tracing beta hydroxybutyrate into citrate, acetyl-CoA, and palmitate. And what we noticed, if you compare your citrate label in both plus and minus lipid conditions, as well as your acetyl-CoA label, you notice that both are reduced. But when you pay attention specifically to the acetyl-CoA label, you see that there's still a lot more acetyl-CoA label getting in as opposed to your citrate. So it tells you that OXCT1 is not required for beta hydroxybutyrate to contribute to fatty acid synthesis, but there's something going on prior to OXCT1 conversion of acetoacetate to acetoacetyl-CoA. The same is for the palmitate label, where the label is shifted to the left. Now notice it is not as drastic as what we observed in the BDH1 knockout, which tells us that there's still some palmitate getting in, even though it is not fully labeling the total palmitate pool. So again, it tells us that even in the absence of OHCT1, there's something else that is allowing for acetyl-CoA to still feed into palmitate. So then it sent us back to literature, and we discovered that there is a cytosolic enzyme that performs the same function as OHCT1 in the mitochondria. This enzyme is called AACS. So our theory is that BDH1 converts beta-hydroxybutyrate to acetoacetate, and this acetoacetate is transported out of the mitochondria, where it is converted by AACS to acetoacetyl-CoA. And we did know that there is a cytosolic thiolase that could convert acetoacetyl-CoA to acetyl-CoA, and that acetyl-CoA can then proceed on to synthesizing palmitate. So again, we knocked out AACS to confirm this, and then we repeated the same experiments, monitoring the growth of our cells in vitro in both plus and minus lipid conditions. Again, the non-targeting control, we still observe rescue when beta-hydroxybutyrate is added to minus lipids. But when AACS is knocked out, we sort of noticed two results, where in one case, beta-hydroxybutyrate did not rescue the growth of AACS knockout cells. Whereas in another, we still see that the growth is there, but not as sufficient as what we observe in our non-targeting control. We then decided to say, okay, let's trace beta-hydroxybutyrate label into citrate, acetyl-CoA, and palmitate like we did previously. And we notice when we compare to our non-targeting control for both citrate and acetyl-CoA, the label drops for both substrates. And you notice that what we initially observed in the acetyl-CoA, especially in either plus or minus lipids, was that the label was not higher than the citrate. So it took away that difference that we had initially observed, which gave us reason to think that the difference that we saw initially, where we had more acetyl-CoA label, could have been as a result of AACS activity contributing to our total acetyl-CoA pool. Similar was noticed with palmitate label. So again, notice it's not a drastic shift to the left, but you still see that there is an effect of knocking out AACS in these cell lines, in that it's the distribution of palmitate label still shifts a little to the left. This is also as a result of still having an active OHCT1. So OHCT1 is still able to synthesize acetoacetyl-CoA, and then other enzymes synthesize acetyl-CoA that are contributing to our current palmitate label. But then we decided to say, okay, so we know that OHCT1 and AACS are able to perform the same function, but what happens if we knock out both OHCT1 and AACS? Are the cells still able to grow? So we did that, probed for both OHCT1 and AACS, confirmed that OHCT1 was knocked out. Now notice there's a band there. I'm not exactly sure what that band is, but I've noticed that this particular antibody for OHCT1 is a bit dirty in comparison to, say, the AACS antibody that comes out very clean. But OHCT1 was still successfully knocked out. It is the band that is more above the other. We repeated our experiments, monitoring the growth of our cells in both plus and minus lipid conditions with beta-hydroxybutyrate, and noticed that when we knocked out both OHCT1 and AACS, beta-hydroxybutyrate did not rescue the growth of either for both of our clones. The same was observed when we traced into citrate and acetyl-CoA. Notice how the label distribution is very similar to what we observed when we knocked out BDH1, where there's barely any label getting into citrate and acetyl-CoA. The same follows for our palmitate label, where the distribution shifts all the way to the left. So that told us that both OHCT1 and AACS are necessary for beta-hydroxybutyrate carbons to contribute to fatty acid synthesis. But then we wondered whether this result that we saw in vitro or in our cell culture would phenocopy in an in vivo setting. So what I did was perform essentially a similar experiment to the mouse experiment that I talked about earlier, except that I put my mice on a controlled diet as well as a calorie restriction diet, because I thought the calorie restriction diet pretty much mimicked our minus lipids conditions better than a ketogenic diet. Because remember, a ketogenic diet is very high in fat, whereas our discovery was that beta-hydroxybutyrate rescues in delipidated conditions. And so my hypothesis prior to this experiment was that we would see a similar result in vivo to what we saw in vitro, where the cells were able to grow in the OXCT1 knockout, but when BDH1 and AACS were knocked out, we didn't see as much of a significant growth. The same follows for the double knockout. But I saw something different. In this case, we noticed that calorie restriction significantly impaired the growth of the OXCT1 knockout. But if you look at our BDH1 knockout, you see that there's basically no significant difference between our control as well as our calorie restriction diet. Similar case for our AACS diet, our AACS knockout. Now, I should note a couple of things. For this experiment, I used the B16s. The B16 cell line is a pain to work with in vivo because the tumors grow really fast. And like I mentioned before, we wait for the tumor to become palpable before we put them on those diets. But the thing is with the B16s, once the tumors become palpable, they take off really quickly. And for the sake of being humane and being ethically correct, we have a limit as to how big our tumors could get. Our limit, we do a bilateral tumor injection, so the combined tumor volume could only get up to 4,000 cubic millimeters big. So if you look at these AACS tumors, they got really big in under 10 days. So you don't really get to see if there really is a significant effect as a result of the calorie restriction diet, just because they take off really fast. So I'm currently doing this in vivo experiment with the AL1376 model. In the previous study that I talked about, they used the AL1376 cancer line, and the tumors, as you saw, took like 20 to 25 days to really grow, so giving you more time to really see if there's an effect of the diet. But either way, this was an interesting result, especially for the BDH1 tumors, because again, my expectation was for them not to grow on a calorie restriction diet. But I then discovered that BDH1 has an isoform in the cytoplasm called BDH2 that performs the same function as BDH1. Now, in the in vitro case, it's possible that BDH2 doesn't perform any function, but my hypothesis is that something about the in vivo environment allows for BDH2 to be activated. So maybe it compensates for the lack of BDH1, which is why we see no difference between our control and calorie restriction diet. I performed the same experiment with our double knockout, our OXCT1 and AACS knockouts. And in this case, we saw a significant difference where calorie restriction diet impaired the growth of our tumors on the CR diet. So that told me that there's something about knocking out OXCT1 and AACS that inhibits beta-hydroxybutyrate contribution to not only palmitate synthesis, but as well as its contribution to cancer cell progression. So based on our results so far, we concluded that one, cancer cells like AL1376s, B16s, and Miapacas preferentially use beta-hydroxybutyrate for de novo synthesis of fatty acids in lipid-limited conditions. Also, conversion of beta-hydroxybutyrate to acetyl-CoA is dependent on its initial catalysis to acetoacetate by BDH1. Beta-hydroxybutyrate contribution to palmitate synthesis can also bypass OXCT1 and citrate production. Additionally, acetoacetate can be shunted into the cytosol to synthesize lipogenic acetyl-CoA through AACS. And the mitochondrial and cytosolic routes through OXCT1 and AACS respectively are the primary paths through which beta-hydroxybutyrate contributes to fatty acid synthesis. So if there's any takeaway from this talk, for one, I had talked about how there are so many diets that are proposed to be cures or treatments for cancer. But what this study shows so far is that, for one, you really have to have an understanding first of how your cancer types respond to specific diets. I had actually done some proliferation assays with the PANC1s, the HeLAs, and the A549s, which I showed don't have the machinery to proliferate in beta with beta-hydroxybutyrate present. But what I observed for those types of cell lines, if you say put a person on a calorie restriction diet, the calorie restriction diet has not been generated for humans, but I could liken it to like a calorie deficit. If a person has those types of cancers and you put them on that type of diet, it may work for them. But say if you put a person who has a melanoma type of cancer, or a cancer that's like your Miyapaka's, on a calorie restriction diet, it may not work for them, because beta-hydroxybutyrate, which our bodies synthesize on low sugar diet, is able to rescue their growth, or they're still able to proliferate. So this study really shows the significance of having some scientific backing to a lot of the literature out there, because one diet may not be a fix-all for every type of cancer. So that being said, I just want to acknowledge my lab, the Leon lab, as well as my lab mates for their contribution to this project, as well as the different cores that we have at VAI, the mass spectrometry core, for allowing us to use their equipment and making it available to us, as well as the vivarium core that supplies and keeps our mice. And other collaborators like Joe Longo. Joe Longo generated our B16 knockouts for us. And Rusty is the dean of our department. He helps a lot with the thinking behind our projects, as well as his input. Just before I open the floor for any questions you guys may have, I thought to also talk about the undergraduate student summer research internship program that we have at VAI. For any of you who may be interested in doing research, you're either interested in going to med school, for example, you want to go to grad school, I strongly encourage you to apply to our summer research internship program. There are 5 research areas at VAI: cell bio, epigenetics, metabolism, and nutritional programming, which is the department I'm in, neurodegenerative science, and structural biology. So there are 5 different areas where you could apply to for the research program. It's a 10-week program, and you pretty much work 40 hours per week. So it's a 9 to 5. You get a feel of the working life, and it's a paid internship. It's also open to international students. The online application opens this December on the 1st, and it closes on February 1st, 2025. I strongly encourage you to apply. I am so grateful for the opportunities that VAI has afforded me. It's given me the opportunity to not only connect with scientists from all over the world, scientists who went to the top schools in this country, as well as beyond. It's also helped me grow very much as a scientist. I started off just like you guys in 2018 as a pre-med major, and I graduated in 2022, international student. I started off wanting to go to medical school. Then I realized in my senior year, I didn't want to do medicine. But thankfully, I had done research with Dr. Lyons, and he could probably tell you I was not the most confident student when I was doing research, but it provided me such a great foundation. A lot of the things that I learned, I first learned tissue culture there. I learned how to look at and count cells there. I learned Western blotting in his lab. So it provided me a great foundation for the work that I do now, as well as giving me guidance over what I really wanted to do. When I realized in my senior year, for that matter, that I did want to do medicine, I recognized that I really enjoyed being in the lab and doing research, and which is what I want to pursue now. I'm currently applying to grad school this year, so please keep me in your prayers. But yeah, I strongly encourage you to seize the opportunity to do a research internship. It is also a great way to make great connections, as well as potentially secure employment for after you graduate. The things that you do now in your undergrad are very important to what happens after this. Especially if your plan is not to say go straight into medical school, you want to take a gap year. The things that you do now set the tone for what you do after you graduate. So seize the opportunity you have, seize the summers that you have to take advantage of things like this. I also encourage you to apply to the research programs within the department, because like I said, it really helped me set a good foundation for where I'm at now. VAI also offers a similar program for medical students to do research during their summer. So if you are planning to go to medical school, or you know somebody in medical school who may benefit from that, it's definitely a great option to take. I also wanted to mention a bit of a shameless plug. I have a YouTube channel. So if you want to gain more information regarding the process of, say, applying to grad school, as well as getting more knowledge about medicine or other fields you can explore in STEM, that is something that I do on my YouTube platform. I also document my own journey with pursuing a graduate degree. So I have a QR code for you. It's easy access. As well as planning to have like guests feature on my channel talking about their various expertise in STEM. I got to learn of a lot of things you could do with research and a degree in biology, which I didn't really know much of when I first started my own journey. So yeah, I will have a lot of people talk on my channel. One of them actually did a summer research internship with us this past year. Following her internship, she also chose to stay with us to work as a laboratory aide. She was working with us even during her senior year. She was able to get some credits off just by working with us. So if that's something that you have the option to do, definitely consider it. So yeah, I had a talk with her on my channel where I asked her questions about her experience doing our summer research internship, and where she shared different factors from the expectations, the things that you do in the lab. It really throws you in the world of research beyond your theoretical knowledge. So I definitely encourage you to check that out. And with that being said, do you guys have any questions? I'm sorry, it's my fault. I talked a lot.
What was your most helpful class, if you could pinpoint one here at Andrews to help you understand what you're doing now?
I would say it was my last class, probably biochem. Biochem really helped me. With just like what I talked about, there's a lot of like pathways involved and things to think about. So biochem really helped me. I took the summer course and it was very intense. So I had to master a lot of the pathways. So even with this, it makes it easier for me to think about different signaling pathways, and just think about the project overall. I think all of them honestly really helped because there's genetics involved, there's cell molecular involved. Scientific communication is also involved because another thing I do in the lab is things like journal clubs where we talk about and read certain papers. I do presentations for work, be it poster presentations or talking to an audience. So different things contributed, but I would say biochem because it was the last class I took and everything was still very fresh in my mind by the time I started working. So yeah. What's up, guys? So I just got back from giving the talk at Andrews University, and a couple of takeaways. First of all, I was very happy and honored to have the opportunity to talk at my alma mater, as well as talk about something that I love, something that I'm really interested in, as well as share with other students the opportunities that are available to them. It was most important to me to be able to communicate the science in such a way that others could understand and be able to interpret the data, as well as where we're trying to go with the research. Yeah, and also bring more attention to the field. It was also important to me to share the opportunities that are available to students that they may not be aware of. So I was very happy and very honored to have that opportunity. It was a big thing for me. It was a very full circle moment. I was a student, just like all the students I was talking to 2 years ago. I sat in those very same seats. I attended research seminars as well. And so it was just very, very nice to get the opportunity to talk to students. Even after the talk, I was able to have lunch with my professors, as well as a couple of students who attended not only the talk, but yeah, I wanted to know about what opportunities could be available to them. So those were like the biggest takeaways for me from that. I really enjoyed again talking to the students after and just sharing my experiences with them. It's something that I enjoy doing. I genuinely enjoy doing, which is why I have this channel in the first place. I really want to share as many resources as I can with you, as well as my own experience, my own personal experiences. So I appreciate when you guys reach out to me, even in the comments or via email or Instagram or LinkedIn, whichever. I appreciate hearing from you, and I try to do my best to help you in whatever way I can, in the best way that I can. So yeah, a couple of different things maybe that I can't remember if I talked about this in the talk. I may have forgotten to mention. So my PI is actually currently looking for a research technician. Well, looking for one for next year. So myself, as well as the other research technician in the lab, or two, are both applying to graduate school this year. So my PI will be looking for a research technician whom we can train to join the lab. So yeah, for those of you who are seniors or know a senior looking to move to Grand Rapids and work at the Van Andel Institute, or looking for an opportunity to work as a research tech, be it for the purposes of like advancing your career for the sake of med school, or if you're wanting to pursue an MD Ph.D, or if you're wanting to go to grad school, or somewhere in between, trying to figure out which direction best suits you, I would strongly recommend you apply, especially if you're graduating next year in May, or if you've already graduated and you're looking for an opportunity to work as a research tech. I strongly encourage you to apply. When the applications open for the job, I will be sure to share that with you so that you guys could apply. It has been a phenomenal opportunity for me to work at the Van Andel Institute. I really enjoy it. For one, it was a great transition for me, coming straight from undergrad and looking for more exposure and opportunity within research, because that's the field that I would like to work in. Also, at the time when I was starting to work in that position, I started working in December. My boss had actually just started that lab in August of the same year. So it was a very new lab, and personally, as I have, come to work in his lab and gotten more exposure to the dynamics of maybe other labs and what it means to work in, say, like a bigger lab, or a lab that has more was established longer versus a lab that was just established or smaller. There are certain pros and cons to both sides that I've observed, but I specifically appreciate one of the pros of working in my boss's lab. First of all, my boss is the best. He has been a great mentor to me, honestly, very down to earth. And yeah, he just has so much expertise in what he does. He is very intelligent. And yeah, he's just been an overall great mentor to have. But also working in his lab, being a new lab, one of the things that I've really grown to appreciate is that, for starters, very new. So when you start a new lab, you also don't have as much funding, or your funding isn't infinite. As a result, you are more strategic about the kind of like projects you do, as well as the kind of structure that you establish. So for me, coming straight from undergrad and going into like the field of research, it was very necessary and important for me to have a certain level of structure, because again, I was not as exposed to the field as say someone who is like a postdoc, for example. So it made it very easy for me to navigate through that, and also learn to establish my own structure, understand my own way of working, and things of that sort. So it's something that I really appreciated. Also, the lab literally started off with myself, 2 other research techs, and the lab manager, and then started to expand from that point. And so I really enjoyed that as well, because working in a smaller lab also allows for the PI to see you more frequently, as well as it also allows for you to just foster a bit more collaboration a little easier, I feel. But yeah, I've really enjoyed it from that point, being able to see my PI on a weekly basis. And just like talk with him about the progress of my project and things of that sort. And even that in itself, as a research tech, one thing that I came to realize in the time I've been working here was that not every research technician in every lab has the opportunity to man their own project. A number of times, like depending on the lab you're in, research technicians at times only like get to assist like grad students or postdocs with their projects, which is fine. But for the direction that I want to take with my life and my career, I really appreciate that my PI gave me a project to man, same as the other research techs, to say, this is your project, this is what I want, and you study from the ground up. And I've really appreciated the opportunity, because it has taught me so much that I'm so grateful for, that I'll probably talk about more in depth in another video. But plus VAI has just been a phenomenal place to work at. It is a very diverse institute. You literally have people from different parts of the world working there. You have some of the smartest scientists who've been to the top schools, both in the US and the world. So it's amazing. It's something that I just never knew that I would get to experience. So the mere fact that God allowed for me to be in this position, I'm always blown away by that, So I truly thank God for the fact that I have had this unique opportunity to get so much exposure. And I strongly encourage you, if this is a field that you want to pursue, or you're even slightly interested in, seize the opportunity. In my last video, I talked about like summer research internships. So if you haven't already checked it out, do check it out. Applications open in December, on the 1st of December of this year, and close on the 1st of February of 2025. So if that's something that you're interested in doing, I strongly encourage you to do that. It may potentially help you with even securing a future employment or future job, just by the fact that you did a 10-week internship, you proved yourself, and you made the right connections. So I strongly encourage you to do that. But yeah, it's been a great time. I'm very excited, and I'm very excited for future opportunities to share my research, to share the work that I'm doing, as well as the work other people are doing with other students. I'm very excited for the opportunity to do that. So yeah, it's been a good time. I'm so grateful for the things that this year has afforded me, the things that God has blessed me with. I think that's another thing that is so humbling about returning to your roots, or returning to like where it all began. There are many times this year and in the past year when I haven't recognized the areas of my growth. Like, I don't really give as much weight to those things. It took me coming back here and being able to talk about what I'm currently doing and interact with my professors as well as interact with students. It took that for me to realize, like, you've really grown. Like, you have truly grown. You're talking differently, you're talking about science differently with a different perspective altogether. You're talking about life differently. This is not the you that you were 2 years ago. This is not the you that probably your professors even recognized or knew. Like, this is not you. This is not that you. You've grown so much. And it's just, it leaves me with this spirit of gratitude and humility, in the sense of like, wow, I can't believe that God did all this for me. Like, I can't believe He opened this many doors for me and has allowed me to mature in this way. So I'm just so thankful for that. It gave me another reason to be thankful today, to just reflect and thank God for how far He's brought me. Sometimes you think only about the things you're going through in the present, the struggles or the challenges that you're facing in a moment. And it's so easy, unfortunately, to overlook the things that God has done for you, the things that He's like brought you through. Sometimes it's so easy to neglect those things, but I'm so grateful for moments like this where you can sit back and just reminisce and almost like just look and say, wow, God, I'm so sorry for the things that I overlooked, and I'm so thankful for the things that You've done in my life, the miracles You've performed, the doors that You've opened, So yeah, I hope you've enjoyed watching this video. I'm so excited for the next guest that I'm going to have on the channel, so keep an eye out for that. I'm looking forward to talking to you guys and sharing whatever resources I can with you guys. Please feel free to message me. Let me know how I can help you. I am more than happy to help. Please take care, guys. I'm praying for you. God bless.